So today we're going to talk about the cell cycle. So our first phase in the cell cycle is interphase, and interphase has several parts. And so we have G1 or gap 1, and that's when the cell grows. And then after cell grows in G1, there's a checkpoint. And the checkpoint is just to determine whether or not the cell has enough proteins to continue on. So G1 or G0, if it doesn't have enough proteins, it goes into G0, which is when it's resting, just normal cell processes. And so after G1, like the checkpoint for G1, is when it's checking for cell size, nutrients, growth factors, and DNA damage. And so also if the cell's big enough again. So if it doesn't, it goes into G0. But if it does, it goes on into synthesis, which is the next phase. So that in synthesis phase, DNA is replicated. And then during the synthesis phase, we have a checkpoint to see if DNA is replicated correctly. So after that, we have G2, or gap 2, is when you're prepping for mitosis. And that's just to see prepping for mitosis so the cell grows some more. And so that's basically it for this checkpoint. For this phase, For G2, before it goes into mitosis, there's another checkpoint. So there's a G2 checkpoint. And that's just to see if the cell's big enough and it has enough DNA to actually split into several other cells. So cell size again, and DNA replication. And so after the interphase is over, it moves into cell division. So cell division is when, it could be mitosis or meiosis. Meiosis is for sex cells. So that's how babies are made and how fertilization occurs. But the first phase in mitosis is prophase. So that's when the chromosomes, chromosomes form. So chromosomes condense into chromosomes and the nucleolus disappears. And in between prophase and metaphase, we have prometaphase. And prometaphase is simply when the nuclear membrane breaks down and kinetochore microtubules, they invade the nuclear space to attach to the kinetochores. And microtubules are polar, so they push against each other and they move centrosomes apart. So that's just when nuclear membrane is breaking down. So after prometaphase, metaphase is now what happens. And so that's when chromosomes line up in the middle of the cell along the imaginary plane in the center of the cell, which is the metaphase plate. So all chromosomes are lined up in the middle. And then we move into anaphase, which is when chromosomes break apart at the centromeres. And so that's when the sister chromatids move apart to opposite ends of the cell. So the anaphase away, okay? And so in telophase, we have when the nuclear membrane reforms, nucleoli reappear, chromosomes unwind, so they break apart back into chromatin like they were in prophase when the opposite happened when they condensed. And then cytokinesis is when we have a pinching of the cell membrane so you can actually see the two daughter cells that are about to form. And so myosin 2 and actin filaments, the filament ring, contracts to break apart the cell into two parts. And so after that, it goes into G0 again, which is resting phase. And then that's just when the new cells are doing their own normal thing. So a kidney cell is doing what kidneys, carrying out normal kidney functions, liver cells carrying out normal liver functions, and then that's about it for that. Okay, so next we're going to talk about meiosis. And meiosis is a little different from mitosis in the sense that sexual reproduction occurs. So this is for sexual reproduction. And so in these, like the first initial cell is diploid, so that means we have 46 single strand like chromosomes. But so let's just note that as 2n because it's diploid. So 2n equals 46. And so DNA replicates in interphase. So that we have DNA replication because it's going to divide. And now we have 2 sets. So we're going to note that as 2 and twice. So that's 46, 46, okay? So, because 2n equals 46. So it's double-stranded here. And we have interphase, remember, this is interphase. And it's double-stranded. Okay, and so meiosis is also different in the sense is that DNA replication happens twice, so the cell divides twice. So the first one is meiosis 1, very simple to remember because first part, and we have segregation. And so segregation is simply when the chromosome number is reduced by half. So segregated, so you're segregating your chromosomes. So homologous chromosome pairs are separated. So we have 2 over here and 2 over here. So 2n became 1n, and since we have 4, 2 and 2, okay? And so in meiosis 2, so like that's just when you have the 2 cells like in mitosis, but it's different. Okay? So after meiosis 1, we only have 2 cells like in mitosis. And then we move into meiosis 2. Is when sister chromatids are separated again. And so DNA copies are separated. So sister chromatids are separated, so they break apart, and then DNA copies are also broken apart. And when this happens, we have 4 cells. So from here, you get 4 cells. Okay, because each cell again divides into 2. So instead of in mitosis where you have 2 diploid cells, so both cells are identical, we have 4 cells in the product. And we're going to note each of these by n because each of these get one of the n's from these other cells. And so we have haploid cells at the end of this. So they're all haploid because half. So they have half of the chromosomes from each. So now they have 23 because half of 46 is 23. So at the end, these all have 23 chromosomes. So that's why we have only 23 pairs because we get one from our mom, one from our dad. And so meiosis is also different because we have this phenomena also called crossing over. So that's when the homologous chromosomes, so let me just draw it like this, and it's when they like come together. And so the little connection that'd be right here would be called the synapsis. So that's when they're paired of the homologous chromosomes. So this is just called the synapsis. And it occurs to non-sister chromatids to increase the genetic variability. So I should have drawn this like with a different color. So they're different. And what happens is part of this chromosome will move over to this chromosome, and part of this chromosome will move over here. So getting your end product to be like this. Let me draw the actual extra part. And then for the other one, you would have this. So instead of it being fully blue, it's going to be partially pink. And so pieces of these pieces of the chromosome, they split, and one of them from this one went over here, and one over this one went over here. So It produces a genetic recombination in the offspring, so the children would have a different combination of traits from both chromosomes, from your mom and your dad. So that's why you get characteristics from each. And so they are joined from the DNA or RNA, could be RNA, but like they're switched and combined on a different DNA molecule. And so like this would be your mom and this would be your dad, and so now part of your mom is over here and part of your dad is on this chromosome. So that's just what crossing over is, and that's a major difference between meiosis and mitosis. Mitosis is just simple cell division, so that's when your organs get bigger, but meiosis is when you're actually creating babies. So this is what happens when like your eggs are fertilized.